UV irradiation, as you know, also causes the formation of another type of pyrimidine dimer, and that is 6,4 photoproduct. So the difference between the CPD and this 6,4 photoproduct is that in cyclobutane pyrimidine dimer there are two bonds between the adjacent thymine residues or adjacent pyrimidine residues. But in case of 6,4 photoproduct, there is only one bond, and that is between adjacent thymine between the C4 and C6 of these thymine bases. Takeshi Todo, Tizi Nomura and their co-workers reported in 1993 that Drosophila melanogaster, which is a fruit fly, has a photolyase that reverses 6,4 photoproduct lesions in the DNA. So they discovered this enzyme 6,4 photolyase in fruit fly, that is Drosophila melanogaster. This photolyase was designated as 6,4 photolyase. It is widely distributed in plants and animals, but has not been detected in bacteria and mammals. So here you can see this is normal DNA, and if it is exposed to UV light, it will make this additional bond between the adjacent thymines. These are two adjacent thymines, and this is 6,4 photo product. And in the presence of 6,4 photolyase enzyme, this is converted back, this bond, this one, this is broken, this is cleaved by 6,4 photolyase, and this damaged DNA can be restored as such. So this is the action of 6,4 photolyase. Considerably less information is known about the mechanism of action of 6,4 photolyase as compared to CPD photolyase. However, the two enzymes seem to work in a similar way. The 6,4 photolyase binds to damaged DNA, as you know, CPD photolyase that bind to CPD. So similarly, 6,4 photolyase binds to the DNA where there is 6,4 photoproduct is present, causing the 6,4 photoproduct to flip out of the DNA helix and into the active site of the enzyme, as in case of CPD photolyase. In the active site of the enzyme, it undergoes a rearrangement to produce a product that receives an electron from an excited FADH molecule. So this mechanism is same as happens in case of CPD photolyase. The photon is absorbed by chromophore pigment, then it is then one electron is transferred to FADH, and then this FADH transfers the electrons to the dimer, and this electron causes the cleavage of bond between the two adjacent thymine bases. The final outcome is that the organisms containing 6,4 photolyase can use light energy to convert 6,4 photoproduct back to normal pyrimidine rings. Organisms can also repair dimer lesions introduced by UV light by excising damaged nucleotides and replacing them with normal nucleotides. This is another mechanism of UV damaged repair. So in this mechanism, the organisms simply remove the or excise the damaged nitrogenous bases. This type of excision repair is the major pathway for repairing UV-induced damage to DNA in organisms such as humans that lack both the types of, that is CPD photolyase and 6,4 photolyase.